Okay, welcome everyone. Good morning or good afternoon or good evening, depending on where in the world you're dialing in from. My name is Nicole Kleinstreyer. I am the director of NICCVAM and the executive director of ICCVAM. And it is my pleasure to welcome you today to our annual Communities of Practice webinar. So I think most of you are aware that ICCVAM promotes the development and validation of methods that protect human health and the environment, while supporting the 3 R's of replacing, reducing, or refining animal use. ICCVAM also provides guidance to method developers and facilitates collaborations that promote the development of new methods. And so this year, the Communities of Practice webinar is going to focus on implementing computational approaches for regulatory safety assessments. And we have some excellent speakers lined up for you. To introduce those experts, I'm going to turn it over to Dr. Helena Hagberg-Durdock from NICCVAM.
Thank you, Nicole, and welcome also from me. I'm really excited for this topic this year, implementing computational approaches for regulatory safety assessment. And as Nicole mentioned, we actually have 3 excellent speakers, and I'm just going to go ahead to take the time and introduce the first speaker, which is Dr. Michael Lowett from US EPA. He is an acting senior science advisor at US EPA, Office of Pesticide Programs, OPP, at the Environmental Fate and Effectives Division, EFED. He has worked there for 15 years, conducting ecological risk assessment in support of regulatory decisions on the registration of conventional pesticides. And he is involved in OPP's efforts with new approach methodologies, or NAMs, and are currently representing OPP on the ecotoxicity work group of ICCVAM and the validation management group of ecotoxicity testing at the OECD test guidance program. And before I'm going to give the floor open to him, I just want to let you know that you can ask questions in the Q&A. And if you have questions for a specific speaker, then just add the name of the speaker. However, we are not going to take the questions until after all the 3 presenters. So you can also ask questions that are more general. So we can ask all the speakers at the same time. And you also have the option to upvote your questions. So if you think that the question that someone else have asked is really of interest for you, just please put the thumbs up, because we take the most upvoted questions ahead of time. And with that, I'm happy to hand over to Dr. Lowett. Please take it away.
First slide, please. Hello, everyone. Thank you to ICCVAM for the opportunity to participate in this webinar. It's these type of events that set the stage for dialogue and building collaboration on advancing the science and incorporating NAMs into regulatory frameworks. As Helena said, I'm Michael Lowett from the Environmental Fate and Effects Division, or EFED, of the Office of Pesticide Programs, or OPP, at the U.S. Environmental Protection Agency. My presentation today is going to be a very high-level overview of EPA's perspective on applying the 3 R's to ecological risk assessments in support of the registration of pesticides, and an overview of one project that we're working on that may lead to reduced animal testing. I'll be focusing on ecological risk, but other divisions such as the Health Effects Division, or HED, have their own efforts. Next slide, please. OPP has a long-standing commitment to the 3 R's principles, which are to reduce animal use. So, for example, requiring fewer whole animal studies, to replace laboratory whole animal studies by the implementation of in vitro studies, which are generally meant to indicate studies with cells or tissues, in chemical studies, which are abiotic chemical reactivity assays. In other words, no cells are used. And computational approaches, also called in silico methods, which include methods like the use of quantitative structure-activity relationships, or QSARs, to predict toxicity. And to refine study protocols to reduce suffering. So, for example, this might be the modification of an existing study design to lessen or avoid animal pain and distress, or it could mean simply requiring fewer individuals in the test. As many of you know, there are other R's that are important to our efforts as well, such as reproducibility, reliability, relevance, and regulatory context. Next slide, please. So if we really step back, an important question to ask is, why would we want to apply the 3 R's principles to the way we currently assess risk from pesticide use? First is for reasons of ethics and animal welfare. Do we really need whole animal testing to be protective of human health and the environment? When is it necessary and when maybe less so? The second reason is efficiency. Theoretically, there should be savings in both time and money once 3R strategies are adopted. A third reason is public health. This gets at the idea that it's known in some cases that test organisms aren't the best models for humans, that in vitro studies with human tissues and associated modeling have greater human relevance and can lead to improved science. A parallel idea for protecting the environment might be using the in vitro studies to test a greater number of taxonomic groups rather than simply relying on in vivo testing with one taxonomic group as a surrogate for other taxonomic groups. And a fourth reason is expectations, which I'll address in the following slides. Next slide, please. Next slide, please. So what does OPP do and why are we concerned with animal testing? OPP is charged with regulating use of all pesticides in the United States. The standard approach is to base ecological risk assessments on toxicity data primarily derived from whole animal studies, which are required in the Code of Federal Regulations, or the 40 CFR Part 158, for the registration of conventional pesticides. Next slide, please. It's very important to recognize that the generation of that data can be costly and uses many individual animals for the registration of a single pesticide. Costs can exceed $100 million, and the number of animals tested can be in the tens of thousands. It's critical that we work with outside partners to advance the application of the 3 R's in a regulatory context. To that end, we are working with a wide variety of stakeholders to systematically look at the studies we require for registration, looking for opportunities to provide waivers to reduce testing and costs without negatively impacting our ability to protect human health and the environment. We're also active in work to advance the use of new approach methods, or NAMs, in regulatory risk assessment, although those activities generally have a much longer time horizon for regulatory acceptance. Next slide, please. So most on this call, but not everybody, may be familiar with the information on this slide. But as part of advancing these efforts, OPP staff are active participants on various committees of ICCVAM, which, as most attending this webinar know, is the Interagency Coordinating Committee on the Validation of Alternative Methods. For those that might not know, ICCVAM is an important stakeholder group consisting of representatives of 17 different federal regulatory and research agencies that require, use, generate, or disseminate toxicological and safety testing information. ICCVAM facilitates the development, validation, and regulatory acceptance of test methods that replace, reduce, or refine the use of animals in testing. My science division in OPP, EFET, is an active participant on the ecotoxicology work group of ICCVAM. The real driving force of ICCVAM is NICCVAM, which is the National Toxicology Program Interagency Center for the Evaluation of Alternative Toxicological Methods, which is part of the National Institute of Environmental Health Sciences, and they provide scientific and operational support for ICCVAM, technical evaluations, and related activities. Next slide, please. So the data requirements in Part 158, which I had mentioned before, provide the scientific basis for characterizing the potential risks associated with pesticide exposure. However, we do have flexibility in implementing Part 158, and OPP has a long history of practicing that flexibility. Additional data can be required, alternative approaches can be accepted, and studies can be waived. Next slide, please. In 2013, guidance was released by OPP on guiding principles for data needs. The purpose is to provide consistency in the identification of data needs, promote and optimize the full use of existing knowledge, and focus on the critical data needed for risk assessment. The guiding principles re-emphasize the flexibility in implementing data requirements in the context of new and emerging tools, which may be used to support risk assessment and risk management decisions. Next slide, please. In 2016, the director of OPP released a letter to stakeholders reinforcing OPP's commitment to the 3 Rs and highlighting 3 main objectives, which are to critically evaluate the studies that impact OPP decisions on pesticide registrations, expand the acceptance of alternative methods, and reduce barriers with stakeholders and other regulatory agencies in the advancement of adopting alternative methods. Next slide, please. In 2019, the US EPA put out a directive from the administrator prioritizing efforts to reduce animal testing. One of the priorities of these efforts is to host an annual conference on the state of the science on the development and use of NAMs for chemical safety testing. As defined in the work plan, new approach methods refer to any technology, methodology, approach, or combination that can provide information on chemical hazard and risk assessment to avoid the use of animal testing. The final reports for the first 3 conferences are available on the website listed on this slide. The 4th conference is currently planned for later this year. Another priority in the directive was the development of a work plan focused on the development, testing, and application of methods that will allow for the reduction in whole animal testing while remaining protective of human health and the environment. Next slide, please. So the EPA's NAMs work plan was created to prioritize agency efforts and resources toward activities that aim to reduce the use of animal testing. This work plan was developed by experts across the agency to set the objectives and strategies for using NAMs. The work plan was updated in December 2021 with the main objectives and strategies being unmodified. Next slide, please. The NAMs work plan identifies 5 objectives for achieving the reduction goals while ensuring that the agency's regulatory compliance and enforcement activities, including chemical and pesticide approvals and agency research, remain fully protective of human health and the environment. One of the useful aspects of the document is that it lays out strategy, deliverables, and timelines for accomplishing the objectives. However, like any roadmap, the work plan represents a snapshot in time. This document will need to continue to evolve as EPA's knowledge and experience grows, and the agency is committed to regularly reviewing the work plan to ensure that the objectives, strategies, and deliverables provide the best possible path to success. Next slide, please. The first of the 5 objectives is to evaluate regulatory flexibility for accommodating NAMs, which seeks to determine how much regulatory flexibility exists for each EPA program requiring toxicity studies and any roadblocks that those programs may have applying NAMs for regulatory purposes. As mentioned earlier, OPP has previously identified that it has flexibility in implementing Part 158 data requirements through waivers and alternative approaches. The second objective is to develop baselines and metrics for assessing progress. This is to determine how many animals are used in a program and track how various efforts have reduced that number. The third objective is to establish scientific confidence in NAMs and demonstrate application to regulatory decisions. Scientific confidence associated with in vivo tests comes from the decades of experience in their development and application. As we transition to use of NAMs for making regulatory decisions, we need to continually build more scientific confidence in the information from those approaches while also establishing the appropriate expectations for their performance and demonstrating their application to regulatory decisions. In part, this means that regulators work with method developers to ensure that NAMs assays and approaches are fit for purpose and have a domain of applicability that is suitable to address the data needs of a specific regulatory context. The fourth objective is to develop NAMs to address scientific challenges and fill important information gaps. In other words, what NAMs do we need to alleviate the need for whole animal testing? The data gaps that need to be filled or the scientific challenge to be addressed is tied to the specific regulatory needs to be addressed. Therefore, a single NAM or combination of NAMs will unlikely be applicable in all need contexts. The final objective is to engage and communicate with stakeholders. I can't stress this one enough. Widespread use and reliance on NAMs agency decisions requires a fundamental change in thinking, given that traditional approaches have been used for decades. Given this, effective engagement and communication with stakeholders is essential to increase acceptance, obtain constructive feedback, and improve the acceptance of using NAMs to inform agency decisions. Next slide, please. So this slide is just to illustrate that there are a variety of strategic plans and roadmaps out there. This slide shows one from ICCVAM and two from EPA. They're generally similar, but what I want to highlight here is that each one indicates that the introduction of NAMs into a regulatory context is an iterative process, which is going to be heavily dependent on the collaboration among stakeholders, specifically the assay or model developers and the end user, such as a regulatory office. Early and continued engagement with the model or assay developers increases the likelihood that a particular NAM can be used in a particular regulatory context. In other words, is it appropriate and fit for purpose for making a regulatory decision? Next slide, please. So now that I've gone through some high-level background, the remainder of the presentation will focus on one project in the Environmental Fate and Effects Division of OPP that may lead to reductions in animal testing by relying on information from an in silico approach. Although I will focus on one effort, we have many ongoing projects, as well as some that have been completed and have led to guidance on potential waivers of studies. For example, in contrast to potential replacement of in vivo data with an in silico approach, we also have retrospective projects, which mean that we're going back through our existing data sets of laboratory studies to determine which of those studies have had the greatest impact on risk assessment conclusions. The idea here is that we might be able to grant waivers for studies that aren't expected to have an impact in the future. Naturally, there may be concerns that using fewer animals in testing could impact our mission to protect human health and the environment. However, it's important to stress that while OPP is committed to reducing animal testing, it is not at the expense of protecting human health and the environment. We will only move forward with any of the 3 Rs once we are satisfied that the alternative is ready for a policy change. Next slide, please. So this slide is to give you a flavor of the types of projects we're working on. The 3 listed retrospective analyses may lead to the reduction in the number of in vivo studies required for these taxonomic groups. The first, on subacute and acute toxicity to birds, has led to a data waiver guidance for avian subacute data based on the results of a retrospective analysis that determined these studies did not change risk assessment conclusions in 99% of the cases evaluated. The fish acute retrospective has been published in the open literature and indicated that tested freshwater fish are typically more sensitive than tested fresh saltwater fish. The next step there is to develop possible data waiver guidance. The Avian Reproduction Project, in partnership with PETA, is very similar to the Acute Fish Project in that the overarching question is, can we conduct a protective screening level risk assessment with whole animal testing on only a single species instead of 2? We are currently completing the data analysis and hope to publish the results as the next step. In terms of replacing required studies, we are considering a QSAR, or quantitative structure activity relationship model, in lieu of acute oral testing with rats. I'll be going more into this project the remainder of the presentation. Another effort led to the potential refinement of the fish bioconcentration study. In this case, it was demonstrated that we could reduce the number of test concentrations and still obtain data acceptable to characterize a fish BCF for certain chemicals. Finally, OPP assesses risk to pesticide transformation products with environmental exposure potential, but ecotoxicity data aren't typically available as it is for the parent compound. To address this uncertainty, we developed a guidance on use of an in silico QSAR model called the Ecological Structure Activity Relationship, or EcoSAR class model, which estimates toxicity for compounds with unknown toxicity based on the toxicity of structurally similar compounds. It is used as a line of evidence for 1, determining residues of concern from pesticide use, and 2, determining the need for requiring additional aquatic organism ecotoxicity testing of transformation products. There is also an ongoing effort by the Office of Research and Development, or ORD, to expand the coverage of pesticide mode of action offered by EcoSAR alone, by extension, improve our predictive capability of this approach. Next slide, please. In collaboration with NICEDM and the Humane Society, the agency is assessing the need for acute oral toxicity to rat testing in support of registration decisions on conventional pesticides, which are active ingredients other than biological pesticides and antimicrobial pesticides. We have been evaluating the utility of the Collaborative Acute Toxicity Modeling Suite, otherwise known as CAPMOS, which is an in silico approach to replace the LD50 values obtained from the acute oral whole animal test with lab rats. Our analysis is complete, and we have submitted our analysis for publication in the open literature. As for all of the other projects I mentioned, the next step is for EPA to determine if the model is suitable for developing a waiver guidance for acute rat oral toxicity studies, which would include under what conditions a waiver could be granted and what assumptions would be made in the risk assessment when a study is waived. Next slide, please. Before we consider the results of our analysis, it is helpful to understand how EPA uses the in vivo acute oral toxicity to rat data in the registration of pesticides. As I mentioned earlier, OPP uses data specified in the 40 CFR Part 158 to make regulatory decisions. For all registered conventional pesticides, an in vivo rat acute oral toxicity test that results in the determination of an LD50 value is performed for technical grade active ingredients, or TGAIs, as well as end use products. We use that data for two main purposes. First, pesticides are assigned an acute oral toxicity category based on the LD50 value, ranging from category 1 to category 4. Those categories determine the precautionary statements placed on the pesticide label for acute human exposure. Second, the data are used to protect wildlife, as the LD50 value is used as a surrogate for acute oral toxicity to all mammalian wildlife. Potential risks to wildlife are determined by comparing a body weight adjusted LD50 value for different size classes of mammals that we are protecting to the estimated acute exposure dose of the pesticide via food items that they may encounter in the environment. Next slide, please. This slide is to illustrate the EPA toxicity categories that are associated with different levels of acute oral toxicity to rats. When these studies are conducted, it's common for limit tests to be conducted that simply demonstrate that the LD50 is greater than 2,000 or 5,000 milligrams per kilogram body weight for compounds that the registrant anticipates are relatively low toxicity. This slide also shows you the type of precautionary statements and signal words that are placed on product labels. Next slide, please. So what is CAPMOS? Most simply, for the purposes of our analysis and this presentation, it is an in silico predictive tool for estimating acute oral toxicity based on molecular structure. This model was built as the result of an international collaboration sponsored by NICEDM. CAPMOS is composed of a suite of consensus models that combine results of individual models contributed by the collaborators. CAPMOS was built with nearly 9,000 chemicals using information on chemical structure and the rat acute oral toxicity data, which was curated from public literature sources. And it was initially tested with a validation set of approximately 3,000 chemicals. Next slide, please. Now that we have an idea what CAPMOS is, the question is, what does it do? CAPMOS can predict endpoints according to a wide range of regulatory needs. For example, binary models. I think we're okay. Binary models, that is toxic or non-toxic, discrete LD50 value models, and categorical models such as the US EPA toxicity categories and GHS toxicity categories. In other words, for our purposes, CAPMOS can predict rat acute oral toxicity endpoints relevant to EPA regulatory needs. That is both the discrete LD50 values and the US EPA toxicity categories 1 to 4. To provide quantitative benchmarks for assessing the performance of CAPMOS predictions, the variability of the reference in vivo data, that is the empirical LD50 values, was determined and is used as a 95% confidence interval around the CAPMOS predicted LD50 values. Incorporating this inherent experimental variability into the CAPMOS predictions helps us build confidence and allows the user to understand the relationship of CAPMOS predictions to the reported in vivo data. Next slide, please. Now that we have a basic understanding of our data needs and the potential utility of CAPMOS to address the data needs, we can consider the project question. On the surface, it is straightforward and can be tested with existing data in the chemical space. Our question is, how well would CAPMOS perform in the chemical space of conventional pesticides in predicting acute oral toxicity LD50 values relative to the in vivo empirical LD50 values of technical grade active ingredients for pesticides registered by the EPA? In other words, can we have confidence in CAPMOS estimates for adequately understanding acute oral toxicity to rats for chemical proposed in the future for registration without the need for an in vivo study? Next slide, please. So how did we do the comparison? We focused the analysis on conventional pesticides registered by EPA from 1998 to 2022, given that more recent chemistries are potentially more representative of future chemistries. We obtained the registrant submitted acute oral toxicity to rat data for the technical grade active ingredient of each of these chemicals. After data set curation, we had 177 chemicals with in vivo data to compare to CAPMOS predictions. The data set curation included considerations such as the removal of rodenticides, because EPA would unlikely grant a waiver for that type of chemical. The final data set had broad and relatively even coverage among herbicides, insecticides, and fungicides with a few additional modes of action. All the data in the comparison data set are publicly available in EPA risk assessments and were conducted according to EPA test guideline. CAPMOS was run using the chemical structures of those 177 chemicals, and we considered the output of toxicity categories and discrete LD50 values along with the 95% confidence interval I mentioned earlier. The accuracy and reliability of those predictions were assessed only relative to the existing guideline in vivo data that EPA uses for labeling and risk assessment. This is an important point because although open literature in vivo data might produce different results and reflect greater variability, the standard used by EPA for registration is the EPA test guideline. Next slide, please. So first, we will look at the results of the toxicity category comparison. This can be illustrated in several different ways as shown in these plots and the table. Ideally, we would like a high proportion of exact matches, and from a regulatory perspective, we want to guard against underpredictions. That is, predicting a chemical is in a less toxic category than would be identified with an in vivo test. The three figures on the left show the relative percentage of matches, overpredictions, and underpredictions on the x-axis for different types of comparison of the data on the y-axis. Exact matches are blue, overpredictions are orange, and underpredictions are gray. The important thing to point out here is the very small percentage of underpredictions in gray, which is what we want to avoid. It's easier to see in the table that most of the in vivo data are in the less toxic categories 3 and 4, and that when those categories are combined, CAPMOS predicts 97% of those chemicals to be in either of those categories. This can be seen in the gray box, which shows that 145 of the chemicals identified as category 3 or 4 with empirical data, shown in the last two columns, were predicted by CAPMOS to be either of those categories as well, as shown in the far left column. 6% of the category 3 chemicals were underpredicted as category 4. From a regulatory standpoint, the overpredictions of category 4 as the more toxic category 3 would be acceptable if it's also acceptable to the registrant. Next slide, please. For the LD50 comparison, it's important to recognize that this CAPMOS prediction is needed for quantitative risk assessment and therefore needs to be more accurate than required for assigning acute toxicity categories. As previously mentioned, CAPMO's predictions of the LD50 include a 95% confidence interval, which is based on variability in published empirical in vivo data. We considered this 95% confidence interval when assessing the accuracy of the CAPMO's estimates for the 177 chemicals in our analysis. Next slide, please. This slide illustrates the result of that comparison. Each of the 177 chemicals is ordered on the x-axis from left to right in terms of decreasing CAPMO's predicted LD50 values. In other words, the most toxic CAPMO's predictions are on the left and the least are on the right. This is shown by the orange bars. The yellow and gray lines show the 95% confidence intervals around those CAPMO's predictions. Paired to each of those CAPMO's predictions is the in vivo LD50 value shown by the blue bar. In those cases, non-definitive LD50 values at either 2,000 or 5,000 milligrams per kilogram body weight are shown as a definitive value on the plot. One obvious observation in this plot is that CAPMOS tends to overpredict the LD50 values. However, from a regulatory perspective, the interesting predictions are in the black box. In those cases, a CAPMOS prediction of 2,000 milligrams per kilogram body weight or higher was associated with an in vivo LD50 value of 2,000 or higher in 96% of the cases evaluated. And the few chemicals with in vivo values less than 2,000 ranged from 1,300 to 1,800 milligrams per kilogram body weight. This is an important finding because there is generally greater room for uncertainty and risk assessment for less toxic chemicals when considered relative to anticipated exposure. Next slide, please. Overall, CAPMOS showed good agreement with empirical values when assigning chemicals to toxicity categories 3 and 4, which represent the least toxic chemicals. But in some cases, the predictions could result in more stringent label warnings if the predictions were used in lieu of in vivo data. A small number of chemicals predicted by CAPMOS to be at the lower or more toxic end of category 3 were in fact category 2 chemicals according to the in vivo data. This highlights the need to consider other lines of evidence to avoid this type of error. Finally, we had too few chemicals in category 1 or 2 to draw useful conclusions, and it's important to note that EPA would want greater confidence in the accuracy of the LD50 values for those types of chemicals, making it less likely that waivers would be granted anyways. Next slide. For the discrete LD50 values, estimates of greater than 2,000 appear to be reliable when considered in a regulatory context. In other words, we can have greater uncertainty about the precision of the estimate as long as we are confident that it is above 2,000. For example, our screening exposure scenario with the highest exposure potential is a small mammal eating food items represented by short grass. In that case, up to 10 pounds of a pesticide, which is a lot, could be applied and we could still conclude low risk potential for a non-listed species if we have confidence that the LD50 is in fact 2,000 or higher. The overall takeaway is that like other NAMs, we don't anticipate CAPMOS to be a one-to-one replacement for the in vivo data, but rather that those predictions could potentially be used in lieu of testing depending on considerations such as the LD50 prediction and anticipated exposure potential. These results will help us inform possible guidance on waivers in terms of under what conditions, which chemicals, and any assumptions that would need to be made in the absence of studies. Finally, additional work could focus on estimating LD50 values for product formulations, which also require acute oral toxicity to rat data, and can show differential toxicity to the technical grade active ingredient because of other included ingredients like inerts and other active ingredients. Next slide, please. In closing, OPP is committed to reducing animal testing burden without compromising the quality of risk assessments. This includes efforts by EFED to lead change in environmental risk assessment. Progress on incorporating reduced animal testing efforts into a regulatory context requires collaboration across many sectors, transparency, and peer review. If you have further questions, you can visit the ICCVAM website or send an email to the address shown. Next slide, please. Finally, work like this can only be done with a collaborative group of dedicated, bright, and creative folks bringing a wide range of expertise and perspective to the table. I'm proud to acknowledge the folks on this team. Thank you for your time and attention, and look forward to your questions during the discussion.
Thank you, Dr. Lewitt. That was really clear and very interesting. And very exciting to see, obviously. So for those of you that just joined, you are watching 2024 ICCVAM Community of Practice webinar, Implementing Computational Approaches for Regulatory Safety Assessment. Please post your question in the Q&A box. If it's for a specific speaker, just put the name ahead, and we will come to these questions by the end of the last speaker. So we have 2 more speakers now. And you can also upvote the questions in case you think that one question is very exciting and you want to hear it answered. So this is bringing us to our second speaker, which is Dr. Pras Padmanathan. And he is a senior scientist at the Office of Science and Engineering Laboratories at the Regulatory Science Research Office within FDA Center for Devices and Radiological Health. His research focuses on credibility of computational models, covering the wide range of models relevant for medical devices, with particular interest in physiological models, including cardiac models. In policy, he led the development of CDRH's first guidance document on credibility of computational models, and he has led or been a key contributor to numerous other initiatives advancing computational modeling in healthcare, including co-funding and co-chairing FDA's modeling and simulation working group, and previously leading OSAL's regulatory science program on credibility of computational models. So with that, please, Dr. Padmanathan, please go ahead and give your talk.
Thank you, Helena. Good morning or good afternoon, evening, everyone. Thanks for the webinar organizers for inviting me to do this talk. So as was mentioned, my name is Dr. Pras Padmanathan. I'm a scientist in OSAL. That's the research science division office within CDRH. That's the devices center within FDA. I'm going to talk about this guidance that we just published called Assessing the Credibility of Computational Modeling and Simulation for Medical Devices. So I want to emphasize at the beginning that this is going to be maybe an outside perspective for those of you in this audience. We're going to talk about how we have decided to evaluate computational modeling and simulation for medical device applications submitted to FDA. And I just want to also emphasize that I don't work in toxicology. So, you know, a lot of this, because this is a very general guidance, some of the concepts and approaches that we use should be applicable to a wide range of models, including types of models that are used in toxicological applications. But I won't be kind of emphasizing that. I'll leave that to the audience to consider what could be relevant to you guys. Okay, I'm going to start by acknowledging my co-authors on this guidance, Kenny Aycock and Brent Craven. The 3 of us together form the the Credibility of Computational Models Regulatory Science Program team members within our office, and they're the people who have worked very closely with me in developing this guidance. But obviously, we had lots of feedback from others in FDA or this devices center at least. The link to the guidance is below. It's very easy to find if you just Google FDA credibility model guidance, or even just FDA credibility guidance. So first, let's talk about what we mean by computational modeling and simulation in the context of this guidance. This figure illustrates that there's a spectrum of mathematical models. On the left-hand side, there are the models where there's a lot of data available, and it's possible to use statistical methods to fit models to the data or use more modern machine learning algorithms. AI kind of generally falls within this category, and they're sometimes referred to as data-driven models, sometimes empirical models. On the other side is the situation where there's a lot of knowledge available about the system, and it's possible to derive governing equations, usually based on physical principles. So here we have what are sometimes referred to as first principles-based models, which includes things like physics-based models or mechanistic models, the image is a picture of Isaac Newton, who obviously postulated that force equals mass times acceleration. That forms the basis of many of the models. But really, this is a spectrum. There's no clear delineation between these two different types of approach. And there are hybrid methods that use both. So for example, first principles based models, physics-based models that have data-driven subcomponents, that will typically be the case that at some kind of level of your computational model, there'll be some, say, material law or and some kind of empirical data required to fit some characteristic of the system that's being modeled. Alternatively, what can happen in regulatory submissions to FDA is that manufacturers develop a first principles computational model and then develop a machine learning algorithm that basically solves that system quickly, trained to the first principle results. So what we mean by computational modeling simulation in the context of this guidance is the first principles based models, either the standalone ones or the first principle part of the hybrid methods, where I'm not going to talk about data-driven models, and the data-driven models are out of scope of this guidance. Next, let's talk about how modeling simulation can be used in medical device regulatory submissions. This slide kind of categorizes 4 different ways. The most traditional and simple example is something that we call in silico device testing, and that's where a computational model is developed of the device itself. So, for example, or generally to evaluate a safety or effectiveness question about the device, which are the 2 types of information that manufacturers need to submit for a medical device to receive marketing approval or clearance. And yeah, in silico testing, a computational model is developed of the device. Some kind of virtual testing is done, and those virtual test results are submitted to CDRH, usually together with other bench, animal, or clinical data. And a completely different case is where, for medical devices, is where a model is actually implemented within the medical device software, or a modeling workflow is implemented within the medical device software. So typically this would be patient-specific modeling. It would be the case that the device takes in patient data, usually imaging data, and then the software generates a computational model of the patient, simulates the patient, and provides the model output to the clinician for use in diagnosis or determining the appropriate therapy. So an example is a device called HeartFlow that takes in, this is basically a software device. It takes in imaging data from the patient, a 3-dimensional geometry of the coronary arteries is developed, and it predicts the level of blockage for certain occluded arteries based on the information taken from the patient and computational fluid dynamic simulations. Third case is something that we're calling in silico clinical trials. And this is a emerging area that there's a lot of interest in, not established yet, but could be a huge development if it becomes established. And basically it's where instead of having just a model of the device, you have a model of the device plus a range of models of patients spanning the indicated patient population, and you can test the device across all of those patients and in some way replicate the real clinical trial. That's why it's called in silico clinical trial. A lot of work in this area, but a lot of questions that need to be addressed before these kind of things could be used on a large scale. And third, the final example is something called an MDDT, that stands for Medical Device Development Tool. That's a separate pathway that we have within CDRH, where instead of the person who's submitting something, instead of them submitting information about a particular device and asking for marketing approval for the device, we do have a pathway for the public to submit information about a tool that could be used to generate safety or effectiveness question about a range of devices. So, for example, that could be a computational model that could be used to address the safety or effectiveness questions around a certain type of device. and we can evaluate those and qualify those. I should have mentioned at the beginning that an example of this in silico device testing is to assess the fracture risk of metallic implants like a hip implant. That's one of the questions that needs to be answered for those types of devices. And typically bench testing is done, but also that is often supplemented by computational modeling, where the solid mechanics of the model of the device is simulated and areas of high stress and strain are predicted. So it's a timeline for how credibility assessment has grown in importance across the last 15 years or so. In the early part of the century, there was growing awareness within our community for the need for formalized credibility assessment. That led to the formation of something called the ICCVAM 40 Committee, American Society of Mechanical Engineers Verification and Validation Committee, which eventually led to the development of this standard, which I'll talk about in following slides. Within my center, CDRH, the Devices Center, we released a guidance document on the reporting and modeling in simulation. And then in recent years, we formalized a research program about credibility of models, and then released this guidance, first in draft form, and then in final form in 2023. So some key definitions before we talk about the guidance. Credibility, we define as the trust based on all available evidence on the predictive capability of a computational model. And we always assess credibility given the specific context of use. We've moved away from terminology such as this is a validated model, and people can just take the model off the shelf and claim it's validated and just use it for any new application. We want models to be evaluated for their context of use, which is the role and scope of computational modeling, answering a question of interest. Then there's two parts to evaluating models: verification and validation. And if we define a mathematical model as basically the equations and the parameters written on paper, and a computational model as a software implementation of that, then verification asks, was this implemented correctly? i.e., were there any software errors? And what is the numerical error? Because we always have to implement some kind of numerical scheme to solve the governing equations, discretizing space and time, and part of verification is estimating what that numerical error. So there's no real-world data, real-world observations needed to do verification, but the real-world data, real-world observations are needed to do validation, which is assessing if the computational model is an accurate representation of the real world. The scope of the guidance, as I mentioned, first principles-based models and hybrid models. For hybrid models, first principle components. Outer scope are standalone statistical or data-driven models, models with no simulation at all, just purely anatomical, such as purely anatomical models. And it's also a very general guidance. So it doesn't provide specific information on how to perform a modeling study or technical details on how to perform the credibility assessment. And it doesn't provide levels of credibility needed for any type of regulatory submission. Because it's so general, that's going to be very case dependent. We built upon the standard that I mentioned earlier, this ICCVAM 40 standard that was called Assessing Credibility Computational Model through Verification and Validation Application to Medical Devices. That provided a risk-informed credibility assessment framework for evaluating computational modeling for medical devices. It was a big milestone for our community at the time. It was the first such standard for medical devices. There's been a lot of interest and use of this, and it provides, as I mentioned, a risk-informed approach. It asks the user to first state what the question of interest is and define what the context of use is, because the guidance is going to use a framework that's based on the same approach. I'll talk more about that in future slides. And then ask the user to define the risk associated with, or assess the risk associated with using the model. And I'll talk more about that in a future slide as well. And then BMB 40 provides these things called credibility factors, which basically break down the verification and validation activities and provides certain features that the user should look into. And it defines those credibility factors. So some key points about the guidance. First of all, it's completely consistent with BMB 40. It builds upon that. It's also risk informed. And there's the same emphasis on question of interest, context of use, and model risk. We'll step through these in a couple of slides in the future. It provides a general framework for evaluating computational models, assessing credibility, and it's intended to be applicable to all first principles based models. So all anything that's not kind of standalone statistical data driven. So a wide range of modeling fields from solid mechanics, fluid flow, electromagnetism, thermal, physiological models of any kind of physiological system, such as the heart or the cardiovascular system. It's intended to be applicable to any kind of application. So all of those applications that I mentioned in the earlier slide, which had 4 different types of ways of using computational modeling for medical devices. And it's intended to be applicable to any type of regulatory submissions. We have a wide range of possible regulatory submissions, from using models to support the initiation of a clinical trial. Sponsors have to, manufacturers have to often provide some data to demonstrate that that's going to be safe for the participants, or sufficiently safe for trial participants, to regulatory submissions for all classes of medical devices, from low risk to high risk. So it's not a prescriptive framework. And it extends the approach of BNB 40. But why do we need to extend BNB 40? Well, the reason for that is BNB 40, when it was written, it really focused on the case of manufacturers generating a model and then validating that model against bench test data using well-controlled, prospective, well-controlled experiments, such as this case, where it's a model of, so basically the question that's being assessed here is when patients have metallic implants, when people have metallic implants, and then they need an MR scan for maybe some unrelated condition, those implants can absorb the radio frequency energy emitted by the scanner and heat up dangerously, possibly. So to understand if it's safe for a person with a metallic implant to undergo MR imaging, computational modeling is often used to assess the possibility of dangerous thermal damage. But if you develop such a model, how can you validate it? It's very difficult to validate that using clinical data. It's often validated using experimental data. So that's an example of bench testing. But we needed a framework that's very general. So all sorts of applications and models and all sorts of different ways of validating it. That's why we developed this guidance, which generalizes BNB 40. This is a figure taken directly from the guidance. Please don't try and read it. It's too small to see, but I just want to emphasize that this is the figure 1 of the guidance. So if you look at the guidance, you can refer back to this and compare it to this talk. This is an easier to read version, and we will go through some of these steps, starting with these initial steps. So first of all, stating the question of interest. The first step is to define what that is exactly, which is the question of interest is the specific question, decision, or concern that's being addressed. And it should be about the real world. It shouldn't be about the model, and it shouldn't be overly broad, such as, is the device safe? So an example related to fatigue fracture, I mentioned that in an earlier slide. That was for a hip implant. This figure is for a stent, but it's the same kind of question. They're implantable devices. An important question that needs to be assessed is, what is the risk of the devices fracturing after a post-implant? So an example question of interest is the device resistant to fatigue fracture under anticipated worst-case radial loading conditions? Second step is to state the context of use. This is defined as the role and scope of the computational modeling in answering the question of interest. So it's a statement on what is modeled, what the key inputs and outputs are, and how the key outputs are going to be used to address the question of interest. Also, kind of critically, it's what other information, bench test data, animal data, clinical data, will be used to answer the question of interest. And I should mention that for our center, when we say bench test, that's a broad term that includes in vitro testing. So an example here is combine, continuing the previous example, combine computational modeling predictions and empirical fatigue testing observations to estimate device fatigue safety factors. So a typical use case is you have a range of new devices, a family of stents, say, and you need to do bench testing, and it can be very burdensome to do bench testing to show the fracture safety for all of the new types of this device, all the different variants. And modeling is used to identify which variant is going to perform the worst, and then only those are examined on in the bench experiment. So it's using them both, but the modeling is used to reduce the experiments that are performed, but then the kind of final decisions are made based on the experiments. So it's kind of using modeling with experimental data. Next is assess model risk. That's defined as the possibility that the computational modeling and simulation results will lead to an incorrect decision that could lead to an adverse outcome. And following or using the approach of BNV40, this is broken down into two factors, something called model influence, something called decision consequence. So first of all, decision consequence is the consequence on the patient of incorrect decision being made. Kind of ignoring the model for the moment, if you answered that question that you're interested in incorrectly, what is the consequence on the patient? Is it minor patient inconvenience? Medium, it could be some kind of clinical intervention is required, surgery. High could be serious injury or death. But this is weighted by the model influence, which is how much are we going to rely on the model based on, but compared to other information. So if we're relying entirely on the model, the influence is considered high, but if we're using the model together to supplement some animal test data or bench data or clinical data, the influence could be low. So the two as a combination are used to evaluate the overall decision consequence. Then the next steps are related to planning the credibility assessment. This follows the approach of BNV40, but we introduced a new concept, something that we call credibility evidence. We define credibility evidence as any evidence that can support the credibility of a computational model. And we provided a categorization of different types of credibility evidence. Now, this categorization is not supposed to be completely comprehensive, though we did try to cover the types of evidence that we see as much as possible. And it's not supposed to in any way be a ranking. It's more of a communication tool. And so I'm going to talk about these first five to give you a flavor of what we mean by these different types of evidence. So the first one is code verification. As I mentioned, code verification is basically testing to ensure that a model has been correctly implemented in software. So basically looking for software bugs. Next one is related to model calibration. Calibration is fitting, tuning, varying parameters so that a computational model matches experimental data in some way. Now, typically this is done as part of a model development process, but a good fit to the calibration data could be generally supplementary evidence in support of the credibility of a computational model, especially when it's a relatively simple mechanistic model that well matches kind of complex behavior. But we emphasize that calibration is not validation. Validation is defined as testing a model against data that was not used in the model development. And the next three steps are different types of validation based on what the model is compared against, whether it's compared against bench test data, whether it's compared against in vivo data. By that we mean either animal or clinical data, or whether it's compared against population level clinical data. And that's the case. That case applies to when we have a range of patient models and it's not possible to validate any particular patient model, maybe because the patient models are generated by sampling. They don't necessarily correspond to any real patient or person out there. So there's no one to compare it to that particular model, but we can compare the range of models as a whole against population level data from the population. So in the guidance, we have this categorization, definitions of each, and some details and device-specific examples for each, and some specific recommendations for each. And the next step, once the user has defined, we don't prescribe what should be submitted for any type of application. We have some recommendations about which kind of to include, but we leave it to the user to define what kind of credibility of the evidence they're going to generate. And the next step, once that has been done, is easiest explained through an example. So let's say that the user is going to generate bench test validation results, then they would go to Appendix 1 of the guidance, which in that section recommends certain credibility factors. And these are factors that break down the evaluation. And at that point, we follow the approach of BNB40. BNB40 defines these credibility factors for this type of test result. So it's things like test samples, the conditions, and it asks the user to generate something called a gradation, which is supposed to be a spectrum of different levels of rigor for each of these considerations, each of these factors. So for a simple factor like test samples, or basically the number of test samples, a gradation could range from a single test sample. So this is, by test sample here, we mean we're referring to the experiments and performed for validation, and we're referring to the number of device samples that are used in those experiments, assuming it's a model of a device. So it could just be a single device sample or multiple samples or a large number. And then the approach of BNB40 is to basically choose a level as part of the prospective planning based on the assessed risk. So if the risk was assessed to be medium, then one of the moderate levels could be chosen from the gradation. So we ultimately end up with a level of credibility that's commensurate with the risk associated with using the model by breaking this credibility assessment down into these different factors and choosing and justifying a level of rigor based on the assessed risk. So that would be, for example, this could be the planned goal of the credibility plan, and you kind of do that for all of these different factors. Then the final steps of our framework, we then ask we recommend that manufacturers do something called adequacy assessment, which at this point it's a prospective adequacy assessment, which is basically answering the question, if providing a justification to say that if all of the above plan is accomplished, justifying why that would be sufficient for using the computational model for the planned question of interest and context of use. And then we have something called a Q submission pathway, that kind of question submission. It allows industry to come into FDA before a marketing application and have us answer questions about their plan. And one thing that we can do is answer questions about a planned model and planned credibility assessment for that model. Then if everyone's in agreement, they can execute the study, develop the valid verification and validation data and credibility evidence, and then generate a final report, including a post-study adequacy assessment. That's what I'll talk about in the final slide, or not nearly final slide, which basically answers the question, does the credibility assessment support using the model for the context of use given the risk assessment? So this is a subjective decision that needs to be made at the end based on all available evidence and engineering clinical judgment. With these types of models, this type of variability in how the models are evaluated, it's very hard to just have kind of quantitative acceptance criteria. We need to still make a subjective decision based on all information at the end, and some considerations such as has everything been tested, and some other considerations listed here. So that kind of concludes the framework. I'm just going to end with a couple of comments about to kind of put this in the bigger picture of what other work that we do in our office. So I mentioned that the standard was developed in 2018, that led to the guidance that came out in 2023. Our office also tries to develop something called regulatory science tools, which are tools that industry could use to support the evaluation of safety or effectiveness of their devices. And some projects that we're working on, developing, because this is a general guidance, what we need are more specific versions of it, or more specific workflows, say, for how that guidance could be used for different types of applications or different types of model. And two important applications are patient-specific models, where models are generated for individual patients and provide information for that patient as kind of clinical tools, or in silico clinical trials, as I mentioned earlier. And we're in the process of developing workflows that follow on from this for those two applications, as well as some other planned activities or continuing activities. Sorry, this is discussed today. I should have deleted that. Related to in silico clinical trials, generating mock submissions that industry could use, or some future potential efforts. This is the webpage for our credibility assessment, regulatory science tools that we develop. And there's a list of these that we've been developing across all of the different regulatory science programs within our office. So, summarize into some key points. Mathematical modeling has a wide range of possible regulatory applications for medical devices, including silico device testing within device software. I mentioned patient models, but this goes very much. This is very closely related to the concept of digital twins and in silico trials. There are many possible ways to evaluate these types of models, so it's challenging to develop a coherent and comprehensive regulatory strategy. But we've addressed this through providing this very general framework that's not prescriptive, and then therefore we provide further assistance to industry through regulatory science tools, but we need to develop kind of more specific standards in the future. Okay, I'll stop there. Thank you for your time, and look forward to questions in the final part of the session.
Thank you very much, Dr. Pratt-Matheson. It was very nice to see also a different application. So again, we are watching 2024 ICCVAM Community of Practice webinar, implementing computational approaches for regulatory safety assessment. We are soon going to go to our last speaker. Please continue, put your questions in the Q&A. If you cannot find your question, it might already have been answered. So then look at the answer tab. And you can also upvote the questions that you find are particularly interesting to you. So our last speaker is Dr. John Arnott, and he's the president and principal scientist of ARC Arnott Research and Consulting based in Toronto, Canada. John is an adjunct professor in the Department of Physical and Environmental Sciences and in the Department of Pharmacology and Toxicology at the University of Toronto. He has more than 20 years of experience in the development, application, and evaluation of databases, methods, and models to assess the exposure, hazard, and risk of organic chemicals to humans and the environment. Dr. Arnott also served on the U.S. National Academies of Science, Engineering, and Medicine Committee on incorporating the 21st century science into risk-based evaluation, and on Canada's Chemicals Management Plan Science Committee. And with that, please, Dr. Arnott, we are very excited to hear your presentation.
Great. Thank you, Helena. And I'd like to thank the organizers of the webinar for allowing us an opportunity to present this work to you today. And I would like to acknowledge my co-authors, some of the members of our research group that are particularly involved with this work. And the title of it is Calculating Bioactivity Exposure Ratios, or BERs, using New Approach Methods for Chemical Assessment. My general objective today is to introduce to the audience various computational tools for applying NAMs in hazard and risk or safety-based contexts. As most people are aware, this is an emerging area of science. Some are very new tools. However, it's important to recognize that some of these tools, like mass balance models, have existed for decades, and the theories underpinning some of these models have existed for centuries. So I'm going to talk a little bit about this, a little bit of the background for the work. And then I'm going to provide a very brief overview of some exposure modeling and tools that can be used for aggregate exposure intake rates. So we can look at comparing in vitro bioactivity data against real world exposures, so we can look at risk and safety. And I will talk about these tools, Protex HT and Easy Suite, which is an online platform of various databases and tools for helping looking at exposure and safety and risk assessment for humans and the environment. The meat and potatoes of the presentation, however, is really in the in vitro, in vivo extrapolation, or IVIVE, to calculate administered equivalent dose, or AEDs. And that, of course, starts with in vitro bioassays, and then in vitro mass balance models, which simulate the fate and disposition of a chemical in an in vitro bioassay. And this has two asterisks here because I'm going to focus most of the talk in this space here, talking about how chemicals distribute within an in vitro bioassay based on their properties and the properties of the bioassay. And then, of course, to extrapolate from the in vitro system to an in vivo condition like a human in the real world, we need to apply high throughput toxicokinetic models or more sophisticated PDK models that look at extrapolating the in vitro dose to an in vivo exposure scenario. Then finally, I will present a case study that brings together these different components, calculating AEDs from in vitro bioassays, looking at aggregate exposure intake rates, looking at exposure models to predict exposures, and then calculating BERs. And this case study is based on some work that we've done recently with colleagues at Health Canada. All right. We're all aware of the fact that there are many diverse chemicals and use contexts that require risk and safety evaluation, and that there's limited information that results then in uncertainty in decision making. And there are various decision contexts, such as chemical priority setting, screening level assessments, and more definitive or comprehensive assessments that usually result in a regulatory decision. And you can imagine then, out of the thousands and thousands of chemicals that require evaluation, one possible strategy to filter through and get to those chemicals that we believe to be of greatest concern is this tiered approach, where one could use margin of exposure or margin of safety or something like that to help filter through. And then we can focus limited time and resources on those chemicals for which we expect the greatest potential concerns to human and environmental health. And this is, of course, an iterative process. It's also, of course, very desirable to have a mechanistic understanding of toxicity and exposure. So, for example, if we have some potential exposures that are approaching or exceeding safety thresholds, we can look at how best to mitigate those exposures if we understand the production to exposure continuum. So we have a mechanistic understanding, and this relates to some of the first principle models that the previous speaker was talking about. All right. And of course, we want to have confidence for different decision contexts in the databases and the models. So this is part of the scientific method where we develop models and we generate hypotheses or predictions of estimates of exposure or toxicity or risk, and then we can ground truth or evaluate those predictions. And there's various receptor contexts, including ecological, occupational or worker exposures, consumer exposures, and the general population. In the case study, I'll be focusing on pretty much this priority setting for the general population. All right, so what is a bioactivity exposure ratio? It is a ratio that links in vitro bioactivity to estimated exposures. So we have on the numerator the administered equivalent dose of a chemical intake rate, for example, in milligrams per kilogram body weight per day, that corresponds to the in vitro bioassay concentration. This is part of the IVIVE extrapolation process. In the denominator, we have an exposure estimate. So this is the real world exposure intake rate. And whether it's using the models that I'm describing today or taking some measured values and estimating an intake rate with an exposure factor handbook, it's always basically modeled. We don't have the capacity to actually measure the intake rates of chemicals into various receptors. So as I mentioned, the first part, calculating AEDs, taking an in vitro effect concentration, applying various tools and models and assumptions then including in vitro mass balance models and high throughput toxicokinetic models to estimate the administered equivalent dose corresponding to the in vitro effect concentration. There are various ways that we can look at exposure estimation, whether it's looking at some monitoring data that's available, or perhaps ideally biomonitoring data. But by and large, for the vast majority of chemicals that require evaluation, there are no or very limited measured exposure data. And it's important to recognize that those measurements are snapshots in space and time, and they might not be reflective of the true comprehensive exposure scenario that may exist. So I always advocate that it's good to look at merging measurements and models together. And when you're in a very data poor situation, maybe all you have is a production volume. And in that case, we can use the models that we've been developing that I'll describe briefly, called the PROTEX HT model, to get an estimate of exposures to humans and ecological receptors. All right. This slide is a lecture in itself, but I think it's very important that people get a comprehensive understanding of the introduction to exposure continuum. All right. Some chemicals exposure occurs at the body, but for the vast majority of chemicals, the exposure process starts way further back. And it's actually can be now quantified from the initial production phases, so developing a chemical, producing a chemical, industrial processes, outdoor use and discard, all of these different processes, part of the life cycle of a chemical, all right, that starts back at chemical manufacturing. Then there, of course, after a chemical is released to the environment intentionally or inadvertently, the chemical is going to move around based on its properties and based on the properties of the environment. And that way we can have chemical fate and transport throughout natural environments and manufactured environments like indoor residences, buildings, cars, and things of that nature. And as we continue through this pathway from production to exposure, there are various pathways of exposure throughout this process, including then various routes of exposure, such as drinking water ingestion, outdoor air inhalation, indoor air inhalation, dietary ingestion through agricultural food items or natural foodstuffs, dermal absorption for chemicals that are applied directly to the body, or in some cases, chemicals that are in our surrounding environment, which partition to our skin, and then there's some potential for absorption into the body. So there's various routes of exposure to our receptors of interest, and we can look at them specifically route by route, or we can total them together to get aggregate exposure estimates. And that's then the total intake rate. And if we use physiologically based kinetic models, we can also then calculate internal concentrations like blood or urine concentrations, whole body concentrations, or tissue concentrations. So this is sort of the exposure continuum. Function of chemical properties, how they're used, environmental characteristics, and receptor characteristics. And it is very important to recognize that this is half of the equation when looking at chemical risk and safety. So a lot of focus goes on at looking at effects and hazard data. But if we want to look at risk and safety-based chemical management, we also need to look at exposure. And as I've mentioned, there are various components throughout this process. There's various models and tools and measurements that can be generated throughout this process from production to exposure. And these tools exist. And what we've been doing recently is then consolidating these tools. And this is a recent paper a few years ago called the PROTEX HT model, which simulates, using first principles, how a chemical moves from its production stages throughout its life cycle, emissions to environments, intentional applications to the body, all these different processes that occur. And we can generate then exposure predictions to humans and ecological receptors in a high throughput context using a first principles mechanistic mass balance approach. And this tool can predict then multimedia concentrations in air, water, soil, sediment, food stuff, indoor dust, all sorts of different components to which humans and others are exposed. And as I mentioned, because we are exploiting the use of PBK models, we can also look at internal exposures like blood concentrations. And because we've developed this model, particularly for data poor chemicals, we've intended to make it relatively easy to parameterize. So we set representative conditions, for example, typical conditions in North America. And then we require only chemical structure, production volume, and functional use data in order to make a prediction of exposures to humans and the environment. I'll show this in more detail in a minute. This is a conceptual figure of the PROTEX HT model used to simulate aggregate human exposure and ecological exposures, which also has the advantage of a One Health approach, so that we're not just looking at ecological and human health separately, but we can look at it in its entirety in a more holistic manner, because it's recognized that sometimes chemicals might be of no risk to humans, but they are to the environment and vice versa. And as I mentioned, we've used the principles of parsimony so that we can make the model relatively parameterization, easy to parameterize with production volume, chemical structure, like a SMILES notation, and functional use category. And what the model is doing then is consolidating various modules that can be used standalone or together in the PROTEX HT framework. We're simulating with the CCCVAM model emission rates throughout the chemical life cycle to indoor environments and to outdoor environments. We're then simulating fate and transport of chemicals between various compartments of the indoor environments and the outdoor environments, including an extensive agricultural food chain and natural food chains for air breathing and aquatic respiring organisms. And then in this case study that we published a few years ago, we focused on the human exposure and with aggregate exposure using these different routes of exposure: dermal absorption, mildly mediated ingestion, for example, hand to mouth or dust ingestion, inhalation of indoor and outdoor air, drinking water ingestion, and dietary ingestion through agricultural food chains, typical of North American humans. And we also have a broad range of ecological receptors, which I can't get into today, but we look at a wide range of representative ecological species. And what the model then provides is estimates of chemical emission rates that can be used to auto-parameterize the systems we're using here, or if you want to take these emission rates and use them to parameterize different models, that is available. It then also predicts chemical concentrations in the multimedia environments, external exposure rates, route-specific intake rates. So for example, if you just want to look at the dietary intake rate to a human or the drinking intake rate, or if you want to look at it all together, you can do that with the model. And you can also look then at internal exposure concentrations like body, blood, and urine concentration. And this is useful because you can then ground truth with biomonitoring data and see if the hypotheses predicted by the models are reasonable compared to independent observations. And when we combine then the exposure information with hazard data, we can get risk and safety estimates. And in this paper, we then showed how we ground truth or evaluate the model. Sometimes the term is validation. In this case, we call it an evaluation. We predicted human exposures to the American population with the model by using only production volume and chemical structure. And we predicted then a broad range of intake rates for a diverse range of chemicals. And after we made these predictions, we then compared with external testing data derived from the NHANES exposures database. So this was work published by John Wamba and colleagues at the EPA. And these are, again, important to recognize, these are estimates of exposures to the American population. But what's encouraging here from our perspective is that with just two basic input parameters, we are able to get very close to what's being observed for exposures in the US population. We aren't fitting the model to these data. We are a priori simulating exposures to the general US population and testing those predictions with independent data. And we can, of course, include estimates of uncertainty in the predictions, and there's obviously variability within the population. But again, this is very encouraging, and we look forward to evaluating the model with further data sets when they become available. But this is pretty much what's available for the aggregate exposure to the US population at this point in time. All right, so that's our little background on exposure. Now, I want to highlight something else that's available here with a broad range of computational tools that are available for free to the multi-stakeholder community. This is the Exposure and Safety Estimation Suite, or EASY Suite is how we like to call it. It's available online. It has a whole bunch of different databases and models and tools that can be applied for exposure and other contexts for hazard assessment. I can't get into this today, but I encourage people to check out this website. It's free registration, and you can start seeing what's available depending on what you may need to address questions and challenges in your day-to-day work. It's a work in progress. It's continuing to evolve, but it's at a point now where we want to share this resource with the community and get feedback and end user feedback. But it integrates curated databases of chemical properties and in vitro, in vivo toxicokinetics, as well as OECD validated QSARs for predicting chemical properties. It includes various environmental fate and transport models that can be used for persistence, long-range transport potential, various bioaccumulation and toxicokinetic models, and then merging all of these tools together to get exposure models like the one I described earlier, Protex HT. And we're just trying to help aid chemical assessments for ecological and human health and chemical sustainability. Now, what Easy Suite is doing really is trying to get these tools accessible to people, but it's also making it easy for people to start using them. Anyone that's had to use a model may know that you have to go look for input parameters like chemical properties, reaction half-lives, things of that nature. And what we've done here is try to make it easier for people to get started. So by simply entering a chemical CAS registration number, structure, or a chemical name into the system will populate the parameters required to run all sorts of models that we've developed, as well as other models that other agencies have developed. And it provides then an opportunity within a matter of seconds for someone to start using exposure and other tools, other models, computational tools. Now, while the system provides automatic defaults for the input parameters, there's also options for people to use preferred information to replace the system defaults. So for example, if the system gives you a log KOW value and you feel that you've got a better log KOW or log P value, you can enter that into the system and replace it and then run the models. And for chemicals that aren't in the built-in database, we allow for various QSAR predictions so that using only a SMILES notation for a new chemical, for example, proprietary chemical, you can then get started right away. And you still have the option then of using user preferred information, chemical information to run these tools. So this is a lot to go through at some point in time, but today I'm just going to focus on the Protex HT model, some of our HTTK models, and the in vitro mass balance model. All right, so getting back to IVE, we need to take our in vitro data and align it with an exposure in the real world so we can then compare with exposure estimates for risk and safety-based calculations. So we've got an effect concentration. Sometimes this could be an initial nominal medium concentration, such as the administered dose into an in vitro bioassay, micromolar. We calculate the concentration at steady state in blood or plasma using toxicokinetic models. And that's basically looking at the terminal or total elimination rate constant. And I'll describe that in a little bit more. And as is current practice, we can assume for conservancy 100% absorption through multiple routes of exposure, although there are capacities within these tools to look at higher tiered information on absorption efficiencies, whether that's through the gut or the skin or through the lung. All right, so we've got our in vitro systems, and we're going to talk a little bit more about in vitro mass balance models in just a minute. That's how we calculate the numerator here, the effect concentration, and then for blood concentrations to extrapolate this to AEVs, we need some toxicokinetic information. And if you don't have measured toxicokinetic data, you can use toxicokinetic models. All right, so in vitro bioactivity assays. These have been in applications for decades on various contexts, but more recently, of course, in toxicology and human safety assessment, looking at high throughput assays and the toxicast data and things of that nature. But it's important to recognize here that this is a system and there are various properties of this system. So for example, we could have an open system or a closed system where there's some air. There's obviously a liquid phase in which the cells are submerged typically. And then there can be additional materials included in the assays, such as the serum albumins and other things of that nature to keep the cells happy. during the tests. And these little dots represent chemicals, and the chemicals distribute through this system as a function of the parameters of the bioassay and the parameters of the chemicals. There can also, in some cases, be sorption or partitioning to the plastic or the sides of the test vessel itself. But what's typically going on is that there's just some system with a volume of liquid and then some mass of chemical added. And that is the nominal medium concentration that's typically being extrapolated to AEDs. However, it's important to recognize that in vitro chemical distribution and disposition within the system is a function of the chemical properties and the assay properties. So depending on your chemicals or your assays, you can get different results in, for example, the freely dissolved concentration. Also, it's important to recognize that in vitro conditions may not be reflective of the in vivo conditions in which they're meant to represent. This is a model. In some models right now for IVIV, it assumes that this medium here is equivalent to blood. And that may be close in some cases, or it may be quite different in some cases. But it's important to recognize that it may be different, and that may have implications in the extrapolation process. So there is a need to put in vitro effects data, the response, into a proper context to inform hazard and risk assessment of chemicals. And that's what we are trying to address here with the in vitro mass balance model. There are various that have been published over the last 20 years. I'm going to talk about some of the work we published about a decade ago. It's referred to as the Armitage et al. Model. What we're trying to do here is to address the fact that freely dissolved concentration, or the concentrations in cells, is not going to be the same for all chemicals in all assays. And it's possible, though, to calculate the distribution or disposition of organic chemicals in vitro test systems based on the chemical specific partitioning properties like log T, log KOW, and the assay specific parameters. So for example, how much serum albumin may or may not be in the system. This model is applicable to neutral organics and many ionizable organic chemicals. I refer to that as IOCs here. And it can include more explicit guidance on parameterization of numerous in vitro test systems. So for example, if you have various well plate characteristics or cell seeding, you can reparameterize the model. It's a generic set of equations which can be parameterized relatively easily if we know the properties of the in vitro bioassay. And that sometimes is frustrating because we can't always find that information or it's not available. But it's important for people to recognize that we know what our test systems are made of and that we know what our chemical properties are, so we can better understand what is happening to the chemical and how that then influences the response observed from these high throughput assays. All right, so as mentioned, chemical is added to the bulk medium, right? We've got our little well plates, things like that. The chemical distributes based on the test system, the assay properties, and the partitioning properties of the chemical. And this, I don't want to overwhelm people with the math, but it's actually a fairly simple set of equations. We have known for over a century that chemicals partition based on their properties. And this is just putting that knowledge into a set of equations that can then calculate the freely dissolved concentration and other concentrations. I'm not going to get into this in any detail. Publications are available and numerous reviews of this topic have been published as well. All right, so what we can do then with the simple EQP model is calculate then the distribution of a chemical within a bioassay. We can predict the chemical mass and mass fractions. And importantly, we can look at concentrations in the dissolved phase, as well as cells, membranes, headspace, etc. And we can then extrapolate these data. So we can calculate the freely dissolved concentration and use blood water partitioning to calculate total blood concentrations. The other thing that this is very useful for, this tool, this computational tool, is that it can identify even before you go and run your test if you may have some issues related to volatility. Maybe your chemical isn't going to stay in here. Maybe it's going to volatilize. Maybe there will be solubility issues and you'll exceed the solubility of the test system and saturate it. So you end up with precipitated chemical in the bioassay, and that's going to influence your interpretation of the results. Maybe there's plastic sorption. Maybe there's so much chemical being added to the system that you get a baseline toxic response, or what's referred to as cytotoxic burst. Important things that can be done before you run your assay. All right, so this is just meant to show in a chemical space, chemical properties span large orders of magnitudes and their partitioning properties for octanol water, air water, and things like this. And as we move from the blue zone, where the nominal concentration is approximately equal to the C-free, we move into conditions where the dissolved concentration may be several orders of magnitude lower than the administered nominal concentration. All right, and this is just again to show you that this is a real factor when it comes to interpreting and understanding the results of your chemical distribution, depending on the properties of the system and the properties of the chemical. Again, published if you want to look at this in more detail. Now, here we'll get into the case study. We've got, for example, a set of chemicals, and here we've got a couple of different bioassays, FG2 and CF7. As some examples, there's differences in the lipid contents in these bioassays. And we've got a set of chemicals. Here we'll start with a relatively low KOW chemical. And as the different cell lines have different compositions and different properties of the chemicals, you're going to see that there's a change here between the nominal concentration, the red circle, and the calculated freely dissolved by the model in the green triangles. Not much of a difference here. We're dealing with relatively water-soluble chemicals. But as we proceed into more hydrophobic chemicals like bisphenol S, bisphenol A, benzo A pyrene, there is a divergence between this assumption that the nominal equals the dissolved concentration or the blood concentration. So here we're looking at in vitro point of departure, different conditions for fetal bovine serum and things like this. And the point is, particularly as we increase hydrophobicity, the assumption that nominal equals C-free is violated, and this can be orders of magnitude difference. All right. So here we get to estimating again, on the log scale, we've got our test chemicals in our case study. Bioactivity based on the nominal versus bioactivity based on the dissolved concentration. This is the bioavailable fraction concentration. And there can be very large differences, particularly for the hydrophobic chemicals. And what the model is doing here is highlighting that the current assumption of using the nominal dose may not be appropriate for different use contexts and decision making. All right, here's another example, basically looking at how we move from in vitro to blood concentrations, and you can see the differences in whether we make an assumption that the in vitro nominal dose equals blood or whether it does not. We use an in vitro mass balance to explicitly quantify then the concentration in various phases of the in vitro assay as well as in plasma or blood. So the extrapolated C blood is the in vitro point of departure for the AED calculations in our case study example here. We've got our steady state blood concentrations, which I mentioned are used or derived from toxicokinetic models. We have doses, we have absorption efficiencies, but the primary driver here is the total elimination rate constant or the half-life. You can obtain this through measured in vivo experimental studies. You can get it from predictions like QSARs, I'll show you in a second, or you can get predictions of biotransformation from hepatocytes or from other sources and parameterized TBK models to characterize then terminal elimination half-lives. I'm going to go quickly here, but there are published and validated models for predicting total elimination half-lives and biotransformation half-lives in humans trained and tested with in vivo data for humans. And here are some of those publications. These tools and models are available in ICCVAM. And we can then use the built-in toxicokinetic models in EasySuite or other platforms for calculating toxicokinetics in various organisms, such as our in vivo models for humans and the other ecological receptors. And these are simple mass balance equations, again, that have been developed and tested for a few decades now. And in EasySuite, we allow people the option to use tiered input parameters from various sources of information if they're available to replace defaults in the system. All right, so now we've gone from our in vitro effect concentrations to blood concentrations, and now applying HTTK models to get CSS, we can extrapolate to administered equivalent doses in milligrams per kilogram body weight corresponding to the in vitro concentrations. And it's different assumptions and different models being applied here. That's why you see different values for the same chemical. And maybe it makes a difference, or maybe it doesn't make much of a difference. It again depends on the properties of the chemical and the properties of the bioassay. We can then further extrapolate then, and here I've compared the extrapolated AEDs with our exposure estimates from Protex HT. We can calculate bioactivity exposure ratios. We can rank and prioritize chemicals based on potential bioactivity. And the results change if you're using the nominal concentration or if you're actually using the in vitro extrapolated explicit mass balance model calculations for C3 in blood. And we can also look then at calculating a BER and internal. So here we're comparing blood concentrations rather than intake rate of exposures. So this is looking at directly in the body, extrapolating from in vitro bioassays to blood, and then comparing those with exposure estimates of blood concentrations using the Protex HT model. All right, so in summary, various models and assumptions can be developed and evaluated for applying in vitro data, recognizing that models are hypothesis generating machines for experimental testing. This is referred to as the scientific method, and it's an iterative approach upon which we can continue to test our current knowledge with independent data. We need to test IV assumptions. In some cases, nominal and blood is okay, but in some cases, can be orders of magnitude different. It's important to recognize when that can happen. And if it's important, then maybe we need to look at more explicit quantification of the IV process. I advocate to anybody that's doing experimental testing or interpreting experimental results to know your chemical key properties and know your assay system, key parameters, because this will influence how different things may be from commonly used assumptions, present state status quo applications of NAM data. While we go through this exercise, in our experience, there is a need for high quality biopartitioning data, so to membranes or to proteins or ionizable organic chemicals. This is a key source of uncertainty in the extrapolation process that is going to take some years in the scientific process for us to improve confidence in understanding the fate and behavior of ionizable organic chemicals in vitro assays and in vivo systems like humans. Again, advocating to be as quantitative and as explicit as possible so that we can align exposure doses across test systems for hazard data comparisons. So when we compare new approach method data with traditional animal testing data, that the doses are in the same units corresponding to the same responses or different responses. And of course, for risk estimation. And one final plug for Easy Suite is that it addresses many challenges that currently exist for better understanding safety and sustainability. It facilitates the application of scientific advancements. We are always updating the tools. It provides public access to many computational tools for chemical evaluations, and I encourage you to try that out. And we are looking forward to working more with stakeholders to improve the system to address specific needs as the science and needs advance. On that note, I just want to thank you again for your time and stop there.
Thank you, Dr. Arnott. Definitely some important things to take in consideration when you're using your in vitro systems as well. So with that, I want to get into the Q&A. And so please continue to post questions in the Q&A if you have them. And I'm going to introduce to you now Dr. Emily Reinke. She's the acting principal investigator at Innoteam, which is a contractor to NICCVAM, and she's going to moderate the Q&A session.
Thank you, Helena. Good morning, everyone. The very first question that we have is actually for the entire panel, and it's from Dr. Cynthia Graham. And her question is that she has concerns about the use of model data as an input into other models. Does this create a less confident answer?
When I go first, I made a comment about this in the text chat, but yeah, I think it can be a concern. It can be unavoidable that the input data for models can't be measured directly and has to be derived from another model. One thing that can be done to mitigate the concerns is uncertainty quantification, where for any particular model, if you can quantify or estimate the uncertainties in the input and then propagate that through the model to get the uncertainties in the output, that gives you some information about how reliable the model is. And if you have models used in kind of series, you can you can take the uncertainty in the first model's output and use that, propagating that through the second model when you use that as the inputs, and then you'll see if the overall uncertainties kind of add up to something that's unacceptable. So that's one way of addressing some of the challenges. One of the goals of rigorous verification, validation, uncertainty quantification is really to quantify the overall uncertainty in model outputs based on all sources of potential error, whether measurement error, whether model form error. And it's a nice approach because it acknowledges that models are flawed in some way, but provides some overall quantitative information on the general uncertainty based on all the different possible causes of error, and that allows the user to decide what to do afterwards.
That's a great answer. Could I just add a little bit to that? And error propagation, Monte Carlo analysis, things of this nature are fairly common techniques used by first principles models. And an important part of that is also a sensitivity analysis. So sometimes you may think kind of parameter in your model going in is really important to the outcome, but it may be completely insensitive. So you might spend a lot of time worrying about, oh my goodness, is the log KW 3 or 4? And it might not matter. So you can do sensitivity analysis to see, does my outcome change at all? As I mentioned in the Easy Suite platform, you can change your input parameters and see how it changes your output there. And that's for these first principles models. With respect to QSAR models and other predictions, there is a lot of work trying to better improve the applicability domain information of a prediction. So is the chemical that you're interested in, is the prediction for a property from a QSAR model within the applicability domain? And that also provides some guidance, although there's not necessarily a unanimous consensus on how best to define applicability domain. I think it's important for end users to be aware that not all predictions from a QSAR, for example, are equal in their predictivity and in their accuracy. And there's a lot of work going on in this space to help better inform what the domains of the applicability of the predictions are and what the uncertainties are. We can also make predictions from a QSAR model and get a predictive interval. And that's where the science is going right now. And then you can take those prediction intervals and put them into a Monte Carlo hallucination, like Pras was saying earlier, and propagate the uncertainty of your input parameters into your model output.
So I'll just add a little bit to, John just said some of what I was going to say. But just to add, even a little higher level from a purely regulatory standpoint, we can do things like we've done for CAPMOs, where we can better define, the domain of applicability, at least in our space, based on how well that model predicts the existing chemicals that we already have, with the idea being that new chemistries that might come in are going to be similar. And certainly that depends on having the data. My office happens to be data rich, so we can do that. That might not be true for another office. And one of the things I alluded to was that just because we put out this publication, that doesn't necessarily mean that's how we're going to implement the results of those models in some way or guidance. I mean, we have to think about, like, where do we have the confidence? So for instance, one thing I can think of is chemicals that have mode of actions that are in the data set that we used as the comparator. We're going to have much more confidence in for a new chemical that hasn't been registered yet, for which we don't have data, that's in one of those mode of action groups, than maybe something that's unique that wasn't in our data set. So that's just another way to kind of less techy get around some of those issues about the uncertainty about the model inputs.
All right, thank you, everyone. Next question is for John. Can you please expand on the Health Canada case study and how regulatory authorities are considering and using computational approaches that you described? And you are muted still.
Sorry. So I can't necessarily speak to that. I mean, we were contracted to work for Health Canada and do a case study so they could start to look at the potential implications of different assumptions that they may or may not be using. I hate to be all political with my answer, but I can't really speak for how these case studies are going to be used at Health Canada, but it's part of the process, right? There's a science that's evolving very quickly. There's excellent scientists in regulatory agencies. And there's a need for some expertise to question and, make some suggestions. And then ultimately it's a stakeholder community, right? And I think with the case study I was showing there, it's again, just showing the fit for purpose aspects of using these tools in various contexts of demonstration. I don't think what we're doing there is going to have any regulatory implications. It's more of just a bit of a build the system, test the system, discuss it, see where it's important, see where we need to maybe look more closely at some things, and big decisions are for other people, not for me. I hope that helps answer.
Yes, thank you very much. So we have another question of what is the range of the difference of the AED, such as order of magnitude between using a normal concentration and the modeled concentration? So this sounds like it's for you, John, again.
So again, what we're showing here is that depending on your assumptions and your models, and I need to stress that the assumption that the nominal concentration is a blood concentration is a model. That's an assumption. It's a model that they're equal. And the in vitro mass balance model is just a different model looking more explicitly at this assumption. And so we have tested the in vitro mass balance model, and we see with the available information we have that it is being more explicit and it's able to better quantify. And in some cases, as I mentioned, it's important because you can get orders of magnitude difference depending on if you're just assuming C nominal equals C blood, or if you're using C nominal and then an explicit mass balance partitioning approach. There can be differences, but there's also a range of chemical space where there are no differences. And one of the contracts we're working on right now is to help better show at a high level where the combination of chemical properties and assay properties may require a closer look, where these two approaches diverge. And all we're trying to do is show in this case study that it can happen, and there's reasonable explanations. We've known how chemicals partition for a century, and yeah, just need to think about things more explicitly in some cases. And again, it depends on the regulatory decision context. For high throughput priority setting, it may not matter that much. If you're getting closer, you may want to look more explicitly at the extrapolation process in a more quantitative in vitro in vivo extrapolation method.
Okay, thank you. And then follow up to both to that, we actually have some questions around the uncertainty again. Either could you introduce an uncertainty factor, or how else would you recommend these predictions with the uncertainty be used in the regulatory arena?
Another excellent question. Good questions from everyone. And again, this is where we're going. All right. So first, we're starting with just understanding there can be differences. We need to test the models. Like I said, the simple assumption that C nominal equals C blood doesn't always work for all chemicals and systems. And where we're currently working with Health Canada is developing a bit of an uncertainty assessment framework for IBIVE. So where we can better characterize what the uncertainties would be expected within a combination of chemical properties and system properties, and where those uncertainty factors may become much larger, particularly for more hydrophobic or volatile chemicals and the conditions that were employed during the bioassay. That also includes, I will add, some of the uncertainty in the toxicokinetics. So for example, when we need to calculate the terminal elimination rate constant, which methods perhaps provide more confidence in applying them in the bigger IBIDE context.
Okay. Any other additions about uncertainty from either Michael or Pros, and how you might recommend handling that?
I'll just quickly, so not specific to that circumstance, but I'll just generically say the way that we handle situations like that. So for instance, uncertainty and a toxicity estimate, we don't apply, at least in my office, we don't apply in EFED, we don't apply safety factors or, you know, whatever you want to call them, right? We don't apply like specific factors to account for uncertainty. But what we do is, since we don't do hazard assessments but do risk assessments, we kind of have it baked in a little bit in that we already have generally conservative exposure estimates. And so we have some, depending on, like, some, just using toxicity as an example, if that's where our uncertainty is, you know, we have some wiggle room there of the relative risk of, you know, whatever our uncertainty is in our toxicity estimate versus whatever our exposure estimate is. So we've got more tolerance, and I think I alluded to that in my presentation. We have more tolerance for that uncertainty, whether we put a number on it or not. The greater that spread is between our anticipated exposure and our toxicity.
Yeah, just a quick comment from me. So UQ at the moment, it's for the kind of applications that we see, it is very burdensome. It's hard to get estimates of uncertainty for a wide range of parameters. It's computationally expensive to do the Monte Carlo sampling with these models that already take a long time to run. And with the risk-informed frameworks that have been developed, there's been a result of that is maybe UQ is kind of considered by some of the community to be only something that's really needed for the really rigorous REQ is only needed for the higher risk applications. And yeah, that may be kind of justifiable, but I hope that in the future, UQ will be more established now, all of our workflows, and as computational costs decrease and tools become more available, because I think it provides a huge amount of information, both in evaluating the model when you do UQ with the validation model, to see how it compares to the experimental data, but also in the final application of the model as well, regardless of what kind of question you're answering. So yeah, it's a big area of research at the moment, and I think it's just going to grow.
All right, thank you all. And John, you just answered that last question I see, so thank you. And that's perfect timing for Nicole to come back in. And again, thank you everyone. And Nicole?
All right, thank you so much. So this has been just an absolutely outstanding Communities of Practice webinar. So thank you all for sticking with us for 2 hours, and thank you especially to our incredible presenters for providing, you know, a wide range of expertise and perspectives on how to establish confidence and encourage translation of computational modeling and simulation approaches into regulatory use. So these presentations have been recorded. They will be available on the NICEDM website soon. So hopefully you are all subscribed to NICEDM News. If not, I don't know how you would have heard about this otherwise. But anyway, you'll get a notification through the NICEDM newsletter when the presentations are available. Thank you all, and have a wonderful rest of your day.